Hello everyone. Today we are going to discuss about the topic nucleoplasm, its composition and function, and nucleolus. First of all, what do you mean by nucleoplasm? This is a colloidal nuclear matrix. It is sometimes called karyoplasm when it is gelatinous or karyolymph when it is colloidal. Nucleoplasm is formed mainly of water, proteins, enzymes, and small amount of DNA, RNAs, etc. Nuclear proteins are synthesized on cytoribosomes. Most of them are contractile proteins. Proteins of the nuclear matrix are of two groups, basic and acidic. Basic proteins include nucleoprotamines and nucleohistones. Protamines remain linked with DNA through salt linkage and histones through ionic bonds. Acidic proteins mostly serve as enzymes. Nucleoplasm contain numerous ribonucleoprotein granules or particles. These probably contain mRNA precursor. Most of them remain dispersed among the chromatin reticulum. They are called interchromatin granules. The others are seen around the chromatin reticulum and hence called prechromatin granules. Nuclear matrix. It is a dynamic system and an active site of enzyme activity. It directs and organizes some of the nuclear activities. It is thought to play an active role in DNA duplication and genetic transcription, and the chemical processing and functional maturation of RNAs and their export to cytoplasm. Nucleolus. What do you mean by nucleolus? A nucleus may contain one or more dense bodies, which is called as nucleoli. They are absent in some algae, undifferentiated embryonic cells, mammalian reticulocytes, spermatozoa, etc. The number of nucleoli in an interface nucleus is species specific and constant or variable. The somatic cell nucleus most usually contain two nucleoli. But in the nucleus of certain amphibian oocytes, there will be 100 or more nucleoli. Nucleoli are very large or very small in those cells which engage in little or no protein synthesis, but large and prominent in protein synthetically active cells. Nucleolus become disorganized during the late prophase of cell division and then gets reorganized during telophase. Nucleolus lack a limiting membrane. Still, it maintains an intact organization due to an optimal concentration of Ca2+. Typically, a nucleolus has four main parts, namely nucleolar matrix, granular zones, fibrillar zones, and nucleolar chromatin. Matrix is the amorphous and enzyme-rich proteinaceous ground system with high concentration of rRNAs and protein. Granular and fibrillar components remain suspended in it. Granular zone is the peripheral part and it contains ribonucleoprotein granules, but fibrillar zone is the central part that contains ribonucleolar filaments. The granules and the filaments of nucleolus remain separate or intermingled. In most cases, the granules are uniformly distributed all over. This gives the nucleolus a homogeneous appearance. In some other cases, they remain clustered in groups, giving the nucleolus a mottled appearance. In still other cases, they are arranged along the periphery, making the nucleolus ring-like. The fibrillar zone of the nucleolus is concerned with the synthesis and processing of rRNA, and the granular zone is concerned with the storage of ribosomal proteins. Nucleolar chromatin exists in various forms. Surrounding the nucleus, there may be ring-like or shell-like layer of chromatin known as perinucleolar chromatin. Often, septa-like processes projected from it to the nuclear matrix. They are called intranuclear chromatin. Nuclear matrix is formed mostly of RNA and protein, whereas nucleolar chromatin is formed of DNA and protein. This nucleolar DNA contains the genes which govern the synthesis of rRNA. Thus, nucleolus serves as the center of the synthesis of rRNA, which in turn forms the precursor of ribosome. Nucleolus organizing center. The formation of nucleolus is usually associated with a specific chromosomal locus, which is called as nucleolus organizer or nucleolus organizing center. It contains clusters or multiple copies of rRNA genes which code for the different type of rRNAs. Nucleolus is in fact a product of chromosomes. It first gets disorganized and then reorganized in each cell cycle. Functions of nucleolus. Nucleolus is the cellular site concerned with the production of ribosomal subunits through the synthesis, processing, and storage of ribosomal constituents. It serves as a site for the production and processing rRNA and also a depot for the storage of ribosomal proteins. In it, these two constituents undergo complexing to form ribosomal subunit. Thus, nucleolus is directly involved in the formation of ribosomes and hence indirectly involved in the protein synthesis. Some ribosomes are the centers of protein productions. Their functions are the following. The first one is synthesis, storage and chemical processing of rRNA. The next one is storage of ribosomal proteins that are imported from cytoribosomes. And the last one is the assembling of rRNA with ribosomal protein to form ribosomal subunits which passes to the cytoplasm, bind together and form ribosomes. Thank you.